Hi everyone, today we'll be talking about the different types of PCR. Before we talk about the different types of PCR, what is PCR? PCR, also known as polymerase chain reaction, is a common technique for amplifying small nucleic acid samples. PCR revolves around the basic concepts of DNA replication involving a DNA polymerase. So now we know what PCR is, but what is actually needed for PCR? To set up a standard PCR procedure, a template DNA with the gene of interest, reverse and forward primers, free nucleotide bases, also known as DNTPs, and Taq polymerase are required. Moving on to how PCR works. It involves three major steps: denaturation, annealing, and elongation. In denaturation, the hydrogen bonds between the double helix are broken by raising the temperature of the solution to 95 degrees Celsius. Next, the temperature is dropped down to between 50 to 65 degrees Celsius for the annealing of primers to the template strand. Finally, in elongation, DNA polymerase starts synthesizing a new DNA strand complement to the template using the free DNTPs in the solution at around 75 to 80 degrees Celsius. Let's take a look at the types of PCR. Number 1. Real-time PCR. Real-time PCR quantifies DNA amplification precisely in real time. It uses fluorescent technology such as DNA-binding dyes or dyes attached to primers to monitor DNA amplification. Special thermocyclers integrated with fluorescence detection modules are used to record the changes in fluorescence. The amount of fluorescent signals is directly proportional to the number of PCR product. By plotting fluorescence against the cycle number, the real-time PCR instrument generates an amplification plot that represents the accumulation of product throughout the PCR. 2. Digital PCR. Digital PCR allows absolute quantification through partitioning the reaction into multiple tiny volume compartments. After amplification, the absorbance of each compartment is counted. 0 indicates negative reactions and 1 indicates positive reaction. The initial copy number and density of target DNA are calculated with Poisson statistics and the number of PCR positive reactions. 3. RT-PCR. Reverse Transcriptase PCR is a combination of reverse transcription and PCR, which generates cDNA from mRNA. The cDNA is later used as a template for PCR. Since RT-PCR uses RNA, reverse transcriptase enzymes and primers of mRNA will be needed in addition to the basic materials of PCR. Before PCR can be initiated, the RNA is denatured and reverse transcribed to create cDNA, which is then used as the template. Thank you for watching till the end, and I hope you enjoyed.